All right. Hi, everybody. Today, photosynthesis. If you've not watched my video already on the process of cellular respiration, I suggest you probably should start with that one because I'm going to refer to that as I go quite often. Today, we're going to talk about photosynthesis, which my former colleague who just retired recently and my former office mate, we shared office for many, many years, Karen Kakiba Russell, states, and she's probably right, photosynthesis probably the most important chemical reaction on the entire planet. And that's because what we're going to do is basically take sunlight energy and convert that into chemical energy. There was a point when we pretty much taught that all organisms on life needed energy that came from the sun. But we have found that there are a couple of unique environments, such as hydrothermal vents at the bottom of the ocean that don't rely on sunlight. But for the most part, nearly all of life in one way or another relies on the energy that comes from the sun and is captured by organisms that do photosynthesis. Most commonly, you would know as plants, but there are a couple others we'll mention as we go. The first thing we need to do is take a little look at what light looks like. So if we take a star such as the sun, or even the lights in the classroom or in your house, we call that light that you typically are used to seeing, we call that white light, and it consists of the entire visible spectrum, which is all the different wavelengths of what we call visible light that you can see with your eyes. Now, using a device such as this here, very simple device called a spectroscope, you can use a prism inside that to divide up the different colors of wavelengths of light that are in that white light. And you've probably seen that before with something such as a rainbow, which basically does the same thing using water vapor. It takes the white light on a sunny day when it happened to rain, and it takes the sunlight from that and using the water and basically splits that into the colors of a rainbow, and those then correspond to different colors and different wavelengths of light. Now, the reason I bring that up there and the reason that's important, and we'll kind of mention this again as we see it, but I'll point it out now so you can kind of see it. But what we're going to see is there's a unique chemical inside plants that give it its green color, and it's called chlorophyll. You probably heard of that before, but there's actually different kinds of chlorophyll, and the interesting part is they absorb different colors within that spectrum, within those wavelengths of light. So plants are green because they actually reflect the green light in the spectrum. So when you have your visible spectrum of what you can see here, okay, so what you see is that right over here in the blue region, and then again over here in the red region, chlorophyll is able to absorb those colors, and that's what it's going to use for the process of photosynthesis. But it is reflecting, which is the opposite of absorbing. It's reflecting the green colors here and here, and so that ends up being what we see. Now, if we sort of take a leaf, if we take a plant, we'll take a look at the leaf, and if we were to zoom in on the leaf, say under our microscope, we would see within the leaf, which probably is no surprise to you, that there are cells in there, very much like what we saw in Alodia would be a good example when we looked at our Alodia under the microscopes, and we saw these plant cells. So if I take one plant cell there, zoom in on that, I would see within that there are a whole bunch of these tiny green structures called chloroplasts, which we've mentioned once before already. And then if you were to zoom into that, which is very hard to do with our microscope, you kind of need a little bit more power than that, you would see that there's a structure within that chloroplast in which there are these tiny little sacs that are called thylakoids. And thylakoids are often grouped together, and when they're grouped together, they call a stack of those thylakoids a granum. And then outside, there's a fluid surrounding the outside area around the thylakoids and the granum, and that's called the stroma. So you can see that also once again here. Here's my chloroplast. So you'll notice it's interesting. There's an outside layer and there's an inside layer. So there's two membranes, which is what we saw with the mitochondria in cellular respiration. And then within that, there's another set of membranes that are very much like plasma membranes that we've talked about before. And those go around each of the thylakoids. And that's going to be important in a second here. So the first thing we're going to do in photosynthesis is we need to understand, well, how is the light energy, how are we capturing that? What's happening with that? Well, the way that works is, and this was discovered in the 1960s by a group of scientists, and what happens is light will come in and it will hit this molecule, and you can see this is my thylakoid membrane right here. So you can see it's a phospholipid bilayer, very much like other membranes we've talked about before. And the light will go in and it will hit this molecule. And what happens is the energy in the light is then transferred to a particular spot within that. And what happens is it's able to use the light energy to excite an electron to a higher energy state. And that's what's going to happen here. Now you'll notice that we have two of these. We do this twice. One's called photosystem 2, and that's the one we start with. I'll mention that in a second. And then we have photosystem 1. So when we look at it, you would think, well, why isn't photosystem 1, over there on the other side, and then photosystem 2? Well, they're labeled this way because this is the order they were discovered. So photosystem 1 was discovered first, but in reality, to explain that whole system, you need to start with what happens over at photosystem 2. So what happens is we excite that energy from the sunlight. We use the energy from the sun, and that energy is transferred throughout this molecule to push an electron to a high energy state. And then we have an electron acceptor, a primary electron acceptor, that's going to take that electron, a protein, and it's going to pass it from protein to protein to protein all the way down until it gets to this photosystem 1. Now you might have seen that before. That might seem familiar. This is a type here of an electron transport chain, although this isn't the one in cellular respiration. This is in photosynthesis, but it's a similar idea. It's basically the same concept where we have an electron at high energy, and then as we drop it, say, downstairs from protein to protein to protein, we're using some of that energy to, once again, which we saw before also, we're using that energy to pump hydrogen ions, in this case, into the thylakoid lumen on the inside of that. So a little bit different direction compared to what we saw in cellular respiration, but it's the same basic idea. Okay. Now, once that electron's gone and you've passed it on, this shuts down. You can't really do this anymore because you've lost that electron. You need to replace that electron. So how do we replace that electron? Well, we do that with an interesting reaction in which we split water. So this reaction is called water splitting. And so this is the reason plants need water in part to run at least the light reactions of photosynthesis, as we'll call them. So we're going to take water, we're going to split the water, and once we split the water, we get from that hydrogen ions, which we're going to use to pump around and do various things. But you notice we also get O2. So you probably know that plants release oxygen, and that's where they do it. It's that photosystem 2. Now, if we go over to photosystem 1 here, light's also doing the same thing, hitting that. It's creating its own electron transport chain, and we're using that energy to generate, as we pass it from protein to protein, we're going to use a molecule that is a low energy state, NADP, and we're going to convert that into NADPH. This might look familiar to you because we saw something like this when we dealt with cellular respiration. You might remember we used NAD and converted that into its high energy state, which was NADH. So we're basically doing that same kind of thing, but it's a slightly different molecule, as you can see there. Now, what happens then is we then use the, as we've pumped those hydrogen ions that I've mentioned, we're then going to have a chemical gradient like we did before. And what's going to happen is they're going to flow through this protein that we've discussed before, ATP synthase. And what does ATP synthase do? It generates ATP from that. And so what we do is essentially we've used that light energy to make ATP. But that's not the most important part. That's a small little piece of the puzzle there. So you might be led to think, oh, that's where it's all going to be at. Not quite. Really what we're doing, this is where the real reaction is important, is making this high energy NADPH, which will come up later on. The ATP is important, but we still have cell respiration to produce that. So we're producing very little ATP here relative to cell respiration. And it's important to note, people always seem to forget this, but plants actually do both photosynthesis and cell respiration. We as humans are only doing cell respiration, but plants are doing both. They do the photosynthesis to generate glucose, as we'll see, but then they use cell respiration to actually make ATP. Here's just another look at it, just another visual of the same basic thing. We call these the light reactions because they require light to run. Here's my photosystem 2, and you can see that it is driven by the P680 refers to the energy level, the wavelength of light that excites that photosystem there. And then over here in my photosystem 1, remember photosystem 2 and then photosystem 1, because photosystem 1 was discovered first, even though photosystem 2 is first in my sort of explanation of it. This is again just another view of that. The thing with visuals like this, it shows you that here's my light energy coming in, and by making a picture where the energy level sort of goes up, you get this sort of gravity appearance of the energy level, which is not exactly what is happening. You know, gravity is not what is driving this. Instead it's in physics of the electron. But I think intuitively many of us understand gravity, and it makes for a good visual. So the light pushes, if you will, this electron up, which is like high energy state, and then if we sort of use the gravity analogy, which again is not really happening here, but visually for us, I think it helps. We're passing it down a series of proteins, kind of like stairs, and each little step is a different protein that releases a little bit of that energy, in that case to pump hydrogen ions, and we make ATP. But again, our goal there is to really make NADPH. Okay, this is a closer view of just a photosystem, what a photosystem is. So you can see that the pigment molecules, the chlorophyll, are scattered throughout this larger structure. And this happens to be chlorophyll A in this case. And basically what happens, the light hits it, and these molecules push and they often call these antenna molecules that will sort of direct that energy to the reaction center, where that high energy electron can be pushed to its high energy state and then be picked up by a primary electron acceptor, and that's then how it passes on. And here's once again just another view of that same thing, showing you that this is actually, here's a membrane, here's my photosystem 2 and 1, same kind of thing. So what we're going to do is do the same thing we did before with cell respiration, but we have 2 steps this time instead of 4. We have first the light reactions. So what are our inputs for the light reactions? Well, the first input that should be obvious is we have light coming in, and we have water coming in. So those are our first 2 key points there. Light coming in. And what comes out of that? Well, O2 is going to come out of that when we split the water. Okay. And we're also going to make this, we're going to make some ATP, which I mentioned before. And remember, anytime you're going to make ATP, that has to come from ADP. So we can put that as our one of our inputs. Most important part is we're taking NADP and making from that NADPH. Where does that occur? This occurs, where's the location of this? Okay, so it takes place across that membrane, the thylakoid membrane. And what's the main goal of this? Well, making ATP is important. Making oxygen is often what students think, oh, making oxygen is important. The oxygen is really like, it's a waste product. We benefit from it, but the plant doesn't care about you. So the main goal really here is to make NADPH. And that'll become more relevant when we go to the next step, which is called the light independent reactions, or the Calvin cycle, named after the person who discovered it, Melvin Calvin. And here's Melvin Calvin in his lab in Berkeley, and he won the Nobel Prize in 1961 for the discovery of the Calvin cycle, which he actually discovered in 1950. So, 11 years. Takes him a while, to figure out who's going to get the Nobel Prize. But Melvin Calvin in his lab, 1961 Nobel Prize winner from UC Berkeley. There he is right there, doing some kind of Calvin cycle stuff. Okay? All right, let's go back to this. Okay, so here is then the Calvin cycle, and let's talk about what happens there. In the dark reactions, we're going to take a molecule that is called ribulose 1,5-bisphosphate here, and we're going to take carbon dioxide. That sounds familiar, maybe that's one of the outputs of cell respiration, and we're going to use this enzyme that we call Rubisco. And Rubisco is going to be our enzyme that allows us basically to attach the carbon dioxide to this molecule that we're building here. And we're going to make these 3 phosphoglycerates, and then we're going to use our ATP up. Remember, we made some ATP in the light reactions, but that really is something we need for this part, okay? And what we're going to do is we're going to use up our ATP. We're taking that energy that is in NADPH, and remember, we made that energy, or we generated that NADPH energy by using the photons in the sunlight and using a special molecule of chlorophyll to capture the high energy photon and push an electron to a high energy state, and then we're harnessing that energy in that high energy electron, and we've captured it in the NADPH. And now what we're doing is we're going to use that energy to generate this molecule here called glyceraldehyde 3-phosphate. If we look back at cell respiration, and even though we didn't memorize probably all the steps, this is one of those early on steps, glyceraldehyde 3-phosphate, G3P. So this is basically a precursor that can be then converted into glucose. And so what we've done here basically in the dark reactions is we've used that to generate our glucose molecule, which now we can use in cell respiration to generate ATP. Now, this Rubisco here, which will be important, which is an important enzyme in the process of the light independent reactions of the Calvin cycle, that doesn't sound very enzyme-like. We've learned we've talked about different enzymes like lactase, and all the enzymes we've seen so far are ending in ACE, right? And you've gotten used to that. Where's the ACE here? Well, actually, Rubisco stands for ribulose 1,5-bisphosphate carboxylase oxygenase. So there's your ACEs. It made up for it. It gave you 2 of them, but that's what Rubisco stands for. So if Rubisco does not sound like an enzyme to you, I don't blame you, but that's because Rubisco is short for that term there. Okay, so let's go through and do the same thing we did for the light reactions and talk about the light independent reactions of the Calvin cycle put it on our chart, figure out what's going to go where, what are our inputs that are important, what are our outputs, where it occur, what's our main goal. And so, first of all, in the light independent reactions, we have this enzyme that we just talked about called Rubisco, and you don't have to learn the long name of it if you want. We'll just call it Rubisco. I think that'll be fine. That's an input. CO2 is an input. And what else have we got here? We've got ATP, that's an input, and we've got NADPH. So then what comes out? Well, once again, if we use ATP, then ADP is going to come out, and if we use our NADPH, that is going to then come out and be recycled as NADP. Okay, not necessarily all that significant, but what's really important, the output here, is G3P. And what I'll do is, the way I like to do it is G3P, and then I like to do glucose, because what you see in all the images almost always is that what you're making is this glyceraldehyde 3-phosphate G3P, but that is one of the intermediate steps in the breakdown of glucose. And so since G3P can easily be moved back into glucose, and to keep the equation sort of nice so you can see it, it's kind of nice to put both of those G3P equals glucose, even though they're not exactly the same thing. They're very, very close, and it just gives you a good idea to sense that what we're doing in the dark reactions, the output that is significant there, is the main thing we're trying to do is produce glucose. And again, that's because plants then use that to run cell respiration to generate ATP. And this occurs inside the stroma. So we'll just add that like that. That's the location. And what's the main goal there? The most important goal, our end product, is we're trying to produce glucose. That part is key for it. Okay, so that's photosynthesis, and I hope that makes sense. And have a good day, everybody. Talk to you later.